common demand from creationists is to provide direct observational evidence of speciation in a laboratory setting. The common issue is that what creationists would consider as speciation tends to be poorly defined, and when it is defined, it tends to be on the scale of what is expected from the results of thousands to millions of years of evolution, if it's expected at all, and obviously not something replicable in a laboratory setting. Speciation has been extensively studied for many years, and there's no question of whether or not it occurs. What laboratory experiments often study are very small and specific parts of speciation, so as to accurately see the mechanisms at work. As a result, creationists tend to dismiss the result of these experiments as not meeting their definition of speciation, or that the very specific conditions that the experiments run under don't represent a realistic scenario that could ever be said to occur in nature. There is, however, an experiment that I've seen referenced that, taken at face value, shows quite a clear and extremely rapid speciation event that would satisfy any reasonable definition of speciation. In 1964, a sample of Nereis worms identified as the species Nereis acuminata were taken from Long Beach Harbor in Los Angeles. The group of 5 or 6 worms were used by Dr. Reich to start a laboratory culture called the lab population. This lab population expanded to thousands of individuals, and over 20 years later, 4 pairs of worms from this lab population were transported to another institution where they once again were bred into a population of thousands. Isolating a small number of individuals from a population creates a genetic bottleneck, often called a founder event. In nature, this can happen when a small population is split from the main group by chance geographical isolation. The new small population will have less genetic variation than the original group, so if an individual in the small population carries an allele that was very rare in the original group, it could quickly become much more prevalent in the new population. While the two populations are isolated from one another, they will be free to drift further apart genetically. This relatively rapid genetic divergence caused by a population bottleneck is known as the founder effect. It was theorized that the founder effect could be a powerful driving force behind speciation, as the differences in the new population could quickly result in them being unable to reproduce successfully with members from the original group. As the Nereis worms were known to have undergone founder events in the laboratory, they became a candidate to investigate the possibility of a rapid speciation event. In order to test this, scientists undertook an extensive search to find wild samples of the lab culture. Nereis acuminata were found at 2 sites, close to where the lab population was originally found, and these samples were labelled as P1 and P2. Then they attempted to mate various combinations of the P1, P2, and lab populations. It was found that while there was reasonable success in mating P1 with P2, neither P1 or P2 were able to reproduce with the lab population. This level of reproductive isolation indicates that the lab population is a separate species to P1 or P2. It was reasoned that since a 5-year search had not found a wild population that matched the lab population, then this was not a species that existed in the wild. And P1 and P2 were said to be representative of what had become of the original wild population because they were the only Nereis acuminata found in the wider area. And since P1 and P2 can interbreed successfully despite such large geographical distances, they were a good representation of the species for the whole area, including where the original lab sample was taken. Ultimately, based on that evidence, it was concluded that speciation had occurred very rapidly in the laboratory after undergoing a founder event. However, after these findings had been published, scientists, including the original paper's lead author, sought to falsify the hypothesis that the lab population had actually speciated in the laboratory, and not before. To do so, they tested the critical assumption that the P1 and P2 populations were in fact representative of the natural population from which the lab population had been taken. They used allozyme electrophoresis to examine the genetic variability within 4 different populations of Nereis worms, and determine the genetic distances between them using Nereis formula. The populations examined were the lab population, P1, P2, and a distinct species of Nereis taken from the Atlantic, as opposed to the other 3 populations that came from the Pacific. The genetic distance between P1 and P2 was very small, as expected, and the distance between the Atlantic species and the other 3 were all similarly large, again as expected. However, despite the lab populations supposedly being more closely related to P1 and P2, the genetic distance between them was even larger than the distance between the lab population and the Atlantic species. If a standard rate of evolution is assumed, the genetic distances can be said to be proportional to evolutionary time, However, as bottleneck events are known to increase the rate of genetic divergence, the distances for the lab population were adjusted accordingly. The finder effects were calculated to account for less than 1% of the genetic distances, so the results remained essentially unchanged. By assuming the average rate of codon substitutions detectable by electrophoresis, as described in the book Molecular Evolutionary Genetics, the genetic divergence between the lab population and either P1 or P2 indicated about 8 million years of independent evolution. A paper was published concluding that it was very unlikely that two bottlenecks and genetic differentiation over a relatively brief 28 years breeding in the laboratory could account for this distance, and the most likely explanation of the genetic distance between the lab population and either P1 or P2 is that they became genetically isolated long before the original lab population was taken. Therefore, the results of the original paper could not support the hypothesis that the lab population had speciated from its original population as a consequence of the finder events during its time in the laboratory. More modern experimentation using Drosophila and high numbers of replicates has established that a finder event, on rare occasions, can induce reproductive isolation rapidly, and while this result might be rare, it did not dismiss its potential importance in generating new species. So, while the kind of rapid speciation event speculated to have occurred in the laboratory Nereis worms can be confirmed to be possible, in fact, reproductive isolation induced by a finder effect and subsequent inbreeding has been observed to happen in less than half the number of generations that the Nereis worms had been bred for. This still does not confirm that it actually occurred, and unless alternative calculations can be provided, this does not address the large genetic distance between the lab population and the P1 and P2 populations. It's possible that the estimated increase in the speed of genetic divergence used to correct for the bottleneck events could now be shown to be incorrect. I haven't seen any actual calculations for this, and if anyone can provide them, I would be interested to take a look. It may be possible then that the vast evolutionary time estimated between the lab and the P1 and P2 populations could be accounted for, or that the evolutionary rate estimations based on the genetic differences can now be said to be unreliable, although I haven't seen anything to indicate that either. However, none of this eliminates the possibility that the lab population was reproductively isolated from P1 and P2 prior to being originally sampled in 1964, since the results would still be equally consistent with a slower speciation event millions of years prior, or a rapid one followed by a long period of little change. The genetic differences between the Pacific and Atlantic Nereis populations were comparable to those between the lab and P1 and 2, and we can reasonably assume that the Pacific and the Atlantic Nereis worms did not speciate recently at all. I suppose another possibility is that allozyme divergence could be significantly more affected by bottleneck effects than the kind of silent DNA differences that is not detected by allozyme electrophoresis. So perhaps another method of assessing the genetic distances between the lab population and its purported parent populations could be formulated. But if this has been done or even proposed, I haven't seen it. Ultimately, the best way to support or falsify the original paper's hypothesis would be to find a wild sample that we can confidently confirm to be representative of the original population that the lab population was taken from. However, I suspect the interest in the original experiment has waned over the years in favour of shorter-term experiments that can allow for a great number of replicates. And while the experiment might be a novel curiosity, I don't think there's much scientific value to be gained at this stage. Incidentally, shedding further doubt on the original Nereis worm paper's conclusion, the lab population has been observed to produce offspring with Nereis worms collected more recently from the same location that P1 was originally collected. And in 2008, worms were collected from the Los Angeles Harbor area that were morphologically identical to the lab population, although I think this was only mentioned anecdotally by Dr. Reich. I hope I've been able to present my reasoning clearly, and unless I've misunderstood or missed something, I conclude that the Nereis worm experiment is not a viable example of speciation being observed to have occurred in the laboratory, and it's a particularly poor example to be brought up in the online debate format. The ambiguity of any positive evidence and the existence of potentially falsifying evidence could easily be used by a dishonest or misinformed debater to cause doubt in the mind of the audience over not only the possibility of speciation, but over any evidence presented from any evolutionary experiments, and the time it would take to dispel such aspersions would eat up a large portion of the debate. All this said, I'd welcome being proven wrong. The Nereis worm experiment would be a straightforward, easily explained and easily understood example of speciation to present if it could be properly supported. And while it might not have much scientific value, such an example does have use in the more layperson's online debate format. Thanks for listening. Any comments or insights would be greatly appreciated.